Kevin received the One Mind Janssen Rising Star Award named in memory of a dear colleague of ours, Dr. Jeffrey Nye, who has been a supporter of this program at Janssen. Kevin, thank you for joining us.
Awesome. Thanks, Eric. So I'm just going to give you a real quick introduction to the research going on in my lab. So our goal is to identify the biological factors that are predisposing individuals to developing major depressive disorder. And so while treating depression is a major goal, it can be very difficult largely because depression is largely heterogeneous. So there are a variety of sort of common behavioral features like hopelessness or sadness that present it in a lot of individuals with depression. There's also a lot of heterogeneity to it. So some patients with depression will sleep more than normal, some will sleep less. Some patients will eat more than normal, and some will eat less. And yet, clinically, these are all classified under the broad umbrella of depression. And so while we know, depression is a very real phenomenon that has very real societal and personal consequences, it's very difficult from a clinical standpoint to treat because we're really lumping a lot of potentially very different things under one category. And so an approach that has been raised earlier by Michelle and others is this sort of idea of personalized therapeutics and the potential for developing approaches for patients with mental illness that can be tailored to the particular symptomatology that those individuals have. And so one of the major challenges in treating depression is that the state of the art is really to prescribe these pharmacologic agents that we know have some efficacy in some patient populations, but there's really no one-size-fits-all approach. That really the general treatment strategy is to try different antidepressants, the serotonin-specific reuptake inhibitors most prominently, and essentially cycle them until we find the one that works. And that, of course, has the problem that there's a lot of patients that don't respond well to these therapeutics, and we really don't know how these really even work on a molecular circuit or on a behavioral level. And so without the sort of understanding of the basic mechanisms of why patients are developing depression, it's very hard to develop these sort of personalized therapeutics that would enable us to treat patients with higher efficacy and fewer side effects. And so our goal then is to understand more about the very basic mechanisms of how these things work. And so in our lab, we're using rodent models because there are a number of things one can't do in human populations. For example, it becomes difficult to establish cause and effect relationships in the brain. And so in the rodent models, we're able to test very specific hypotheses about the roles that specific molecules or cell populations play in the depressive behaviors that we're interested in. Of course, the issue is when one uses a rodent model is what exactly is rodent depression? Unlike humans, where we're able to communicate how we feel, and certainly that's one of the major ways that depression is communicated, rodents can't really do much of the same. And so we're reliant on interpreting their behaviors. But in effect, this might be a sort of blessing in disguise because we have to provide a laser focus into the behaviors that the rodents are showing, and we can therefore start to understand these very basic mechanisms of the behaviors, the symptomatology that is associated with depression. And so rodents, like humans, respond to chronic stressors in ways that are not always adaptive that can lead to symptoms associated with depression. And so using these rodent models of chronic stress, my lab is proposing to investigate the biological sources of depression vulnerability. And so there is some discussion and comments about the identification of biomarkers for depression, trying to identify individuals who are vulnerable to developing depression before they do so. And so the goal of our lab is to actually identify in the brain what these biomarkers may be early on before depression develops. And then once we've done so, because we're using a model that has very high resolution, we can then dive more deeply into developing potential pharmacological interventions for treating these patients. And then, of course, these can be used potentially down the line in combination with the sorts of behavioral tracking tools that Michelle was talking about, to target these for particular patients suffering from mental illness, to push forward this idea of personalized therapeutics.
Thank you, Kevin. Let me follow up with a question. Michelle talked about the importance of anhedonia as a very difficult symptom of depression syndromes in humans. You talked about trying to study depression-like phenomena in a mouse or a rat, which can be challenging, as you acknowledged. How can you tell if a mouse or rat gets depressed? But anhedonia is actually a type of symptom that perhaps we can study quite well in a mouse or rat. Can you say a little bit more? What are the kinds of things that we would do to see whether chronic stress in your hands would make a mouse or rat experience an anhedonia-like phenomenon?
That's right. So there are a number of behaviors that one could study in the rodent model to look at anhedonia. For example, we propose to use this social defeat stress model that your lab has used quite prominently. And one of the behavioral features that some of these animals develop is this anhedonia phenotype, which in rodents is often expressed by what's called a sucrose preference. And so we can identify animals that essentially don't feel the same reward from drinking sugar water that normal animals do. And there are also a variety of other similar sorts of tests that can identify animals as undergoing anhedonia or not. And so our goal is to link, using the mouse model that we're using, essentially how the animal responds on an activity level in the brain to whether they develop this anhedonia phenotype or not.
Excellent. And I think it's important to emphasize that a mouse or rat, just like a human, enjoys doing things in its environment, whether it's eating Reese's Pieces or drinking sugar water or having sex or having social interaction. And we can measure the degree to which a rodent participates in those activities that are naturally rewarding for it, just the way we as clinicians evaluate how patients with depression are able to enjoy themselves through their own rewarding activities. Thank you very much. One other question before we move on to the next Rising Star Award recipient. One of the features of your proposal that the review committee commended was its use of an unbiased approach. And I was wondering if you could tell us what that means and why it's so important.
Yeah, so one of the major limiting factors of our investigation into the sources of mental illness is that inherently the approaches that we're using are biased by the results that come before them. And so this makes sense because we're building on the discoveries of others, but it also funnels us into very specific avenues that we may be missing the proverbial forest through the trees. And so my approach that we're taking in the lab, it is using some of the foundations that others have built, but we aim to cast a much broader net to come in with not a specific idea of how the brain is working or what regions, cell populations, molecules might matter, but to essentially listen to what the mouse is doing, look at what our readouts are telling us, and use the data to then select the major brain regions, major cell types that are driving the behaviors of interest. So hopefully we can discover new things and basically accelerate progress towards development of new therapeutics.